Hello, biologists. We're going to talk about diffusion, osmosis, and glucose uptake. So the first thing I like to do when I think about diffusion osmosis is think about how things are moved in general. So I've got this nice flow chart for you. On this flow chart, we can start with the idea of movement of substances, and we can break it down into two groups. Are you moving something small, or are you moving something large? If you're moving something small, is it something that is not going to require energy? That would be called passive. So passive transport, no energy required. Or is it something that will require energy? That's active transport. Now you might be like, wait, don't those apply to large substances? In general, if it's large, it's gonna be active. So I would go ahead and call everything on this side active transport as well. That is me trying to spell the word active using my computer mouse. So active transport. In a large substance, you're either going in or out of the cell, and that's going to take a lot of energy. You're going to take one of your little vesicles, your transport vesicles, which are made of phospholipid bilayer, and they contain something keeping it separate from the cytoplasm around it. And if you take it into the cell, it's called endo for inside, cytosis for cell. So endocytosis is moving something into the cell. You can either move in a solid or you can move in a liquid. If it's a solid, it's phagocytosis. And if it's a liquid, it's pinocytosis. I remember that because I think Pinot is a type of wine and that's a liquid. So if you're taking in Pinot, it's pinocytosis, you're taking in a liquid. If you're moving things out of the cell, it's going to be exo for outside cytosis. All of that is a lot of energy. So it will require ATP. So this could like fall under this umbrella if we wanted to add that arrow there. If your movement for a small molecule does not require ATP, we have two things that we think of. Osmosis, which is the movement of water specifically, so that's H2O, and diffusion, which is the movement of everything else. So that's going to be ions, molecules, gases, the large things, small things, everything else. Now, osmosis and diffusion both follow a guideline where they're going to move from a high concentration to a low concentration. You might be asking yourself, what's a concentration? Allow me to give you a visual. This is a concentration. We have a lot of blue dots on one side and very few dots on the other side. Concentration in this case refers to the density of something, and a gradient means from going to high to low. So this is a concentration gradient. In diffusion, your ions, your molecules, maybe it's little bits of tea that I put a tea bag in a cup of water, or it could be the smell of cookies cooking, whatever it is. It's going to go from high concentration to lower concentration until it's equally spread out. So if you imagine you're baking cookies, and you have cookie dough cooking in the oven, the reason that you can smell it permeating around the house is because the high concentration of the odor particles are coming out of the cookies as they cook, and those molecules of smell are bouncing into each other like billiard balls. And so they knock into each other and they go away from each other after they bounce into each other, and then they space out because of that. That bouncing into each other is referred to as Brownian motion. So everything in existence, even if it looks like it isn't moving, it's just kind of jiggling. And it's jiggling and jiggling and jiggling, and two things will eventually jiggle together, knock into each other, and then bounce further apart because of their Brownian motion. So diffusion, both solute and solvent move. Let's talk about those vocab words. Solute is the item that is being dissolved. In this case, it's the red dots. It would be the smell of the cookie or the tea in my tea bag. And then solvent is the thing that dilutes the item that is in it. And so the solvent here would be this water solution could also be oil, could be air. Whatever the thing is that it's dissolving into is the solvent. Now, osmosis is a type of diffusion, but it's the diffusion of water, and specifically through a semi-permeable membrane. So this won't happen if there's not a semi-permeable membrane. Here I have a beaker, and this beaker is divided with a semi-permeable membrane. A semi-permeable membrane means that some things can go through it, but not others. Often it's size dependent, so there's small holes. Small things can go through, but big things can't. Think about like a screen on a window. Air is allowed to go through, but bugs cannot. Or it could be charge dependent, or it could be controlled by a living cell. And then maybe the cell decides that it will let in water, or it'll let in glucose, but it won't let in, say, salts. So your semi-permeable membrane allows some things through, but not everything. Semi and permeable means going through. In diffusion and osmosis, we're always going to go from high concentration to low concentration. So just as these molecules spread out, these water molecules are also going to want to spread out the solute. And so here, where there's fewer molecules, there's more free water. Here, where there's lots of molecules, there's less free water. And so the water will move from high water concentration to low water concentration. It's going to move until it spaces out those molecules so that they are equally distributed on both sides. Now it looks like water is moving from low solute to high solute, and that's true, but what's really happening is the water is moving from high water to low water. So let's practice thinking about this. Anywhere that we're going to have more solute, we're going to call it hypertonic. Hyper means more, and tonicity refers to the solute level. So hypertonic means more solute. If you have less solute, it's hypotonic. So think if you are hyped up, you are excited, you have more energy. If you're getting a shot, you're using a hypodermic needle. It goes below your dermis. So hyper above, hypo below. And if you have equal amounts of solute, then we use the prefix for equal, which is iso. So we have hypertonic, lots of solute, hypotonic, little solute, and isotonic, equal solute. In this practice question, we have our solute, sodium chloride, that's table salt, and one side of my semi-permeable membrane has more. There's more sodium chloride on this side. So this side here is hypertonic. The other side has less. It is hypotonic. Because of that, there's more free water on the side with 20% sodium chloride. Why is that? Because if it's 20% sodium chloride, then the other part of it will be, oops, sorry. Go back one. There we go. If it's 20% sodium chloride, the other part of the solution, the 80%, is going to be water, because we have to have 100% total. So that's a lot of water. Here, if it's 50% sodium chloride, that means that there is 50% water. So where is there more water? Where there's less solute. Water wants to move from high concentration to low concentration. So water will move through the semi-permeable membrane into the side with more solute until the solutes are equally spaced out. Let's practice the next one. Use your osmosis vocab to describe the relative concentrations. So identify what they are and then which way the water will move. Our solute concentrations are the same, which means they're isotonic. Because they're isotonic, the water will move equally in both directions. So it'll move both ways. Here, now we have a cell in a beaker. Which way will the water move? And what will happen to the size of the cell? There is more solute inside the cell. So the cell is hypertonic to the solution outside. The solution outside is hypotonic to the cell. The water will move from high concentration to low concentration. So the water is going to move right into this cell. And think about filling a water balloon. What's going to happen to the size of the cell? It's going to increase. Here we have a cell with no solute in it, and we have a solution that's 40% sodium chloride. Which way will the water move? What's going to happen to the size of the cell? And use your osmosis vocab. This has more solute, so it is hypertonic. This has less solute, it is hypotonic. The water is going to move to wherever there is more space for water molecules. So water moves from high concentration of water to low concentration of water. You can also just think water moves to wherever there's more solutes. And so this cell will start to lose water and it will shrink. It will decrease in size. Here's a practice question for you. Your rebellious 13-year-old cousin is about to pour salt onto this beautiful Pacific banana slug because she saw a cartoon of someone doing it. She doesn't understand that salting the slug will kill it, and luckily you stop her just in time. Explain to your cousin what will happen to the slug if she salts it using your osmosis vocab. If I have 100% salt on the outside, that's 100. Do you think my slug is composed of 100% salt? No, it's going to be less. So whatever it is, let's just say it's 30% salt, because that feels like an awful lot of salt to be inside of a slug. So where is it hypertonic? It's hypertonic outside. It's hypotonic inside. Which way will the water move? The water will move from high water concentration to low water concentration. That means that it will move from low solute to high solute, and your little banana slug is going to get severely dehydrated very rapidly, and that would unfortunately kill this beautiful banana slug. So now that you've explained it to your cousin, she goes, oh no, and she leaves it alone and decides that she loves banana slugs and maybe goes on to study them in her life. In plant cells, we also really care about osmosis. I actually have a plant to show you. If your plant is happy, it likes to be full of water, and it prefers being in a hypotonic solution because of that. Why? Because if it's in a hypotonic solution, that means that the water will be moving into the plant cell, and the plant cell will be full. It'll be storing the extra water in its central vacuole, and it'll be nice and plump. Cells like this have high, what we say, turgor pressure. They're turgid. Plants prefer high turgor pressure. If your cell is in an isotonic solution, it's gaining and losing water at the same rate. And so it'll be okay, but it won't be plump. It won't be crunchy. It won't be juicy. It won't be super full of water, which is what plants like. And if your cell is unfortunately in a hypertonic solution, there's more solute outside, so there's less water outside. Your plant cell will lose water, and it can decrease the volume of the central vacuole so much that it actually pulls the cell membrane away from the cell wall, and it can totally dehydrate. We call that like lysis or crenation, where it just sucks itself in, and it will absolutely kill your plant cell. I have two of my peace lilies here. This one is pretty happy. It's well hydrated. It's not looking like super thrilled, so I would say that this is in an isotonic solution. I need to water it. But peace lilies are also super dramatic. So here is another peace lily. This one is very sad, very dehydrated. This is in a hypertonic solution. I have very limp, placid leaves, and so I'm going to soak this in the bathtub today. But you can see how dramatic plants like to get when they get dehydrated. So this is a plant with low turgor pressure, and I will be taking care of it after I film this video. The same thing is really important for us in our red blood cells. So when you have a red blood cell, you want it to be that perfect biconcave disc shape that we can see right in the center. It's got that dip in the center. It's nice and round. It's got an increased surface area with that dip. Its shape is related to its function. If you put a red blood cell in a hypertonic solution, it'll start to lose too much water and it will shrivel and it will become non-functional and it will die. So you do not want too much solute in your bloodstream because that will cause your cells to shrink. Our vocab word again, crenation, means shrinking of those cells. If you put a hypotonic solution in your blood, there's not enough solute. That means that there's more solute inside your red blood cell. You'll be like, oh, cool, they hydrate. But you don't want your red blood cells hydrated. You want your red blood cells to maintain their functional shape for carrying oxygen. They don't care about carrying water. They care about carrying oxygen. So if too much water enters your red blood cells, they pop and they explode and they become useless. And that can be fatal. I want you to take a moment and think about 3 more examples of diffusion or osmosis in the human body. We have a lot that depends on diffusion and osmosis. The first thing that I think of is your lungs. Your lungs function to suck oxygen out of the air, but the whole way that they function is just diffusion. So every time you suck in a big breath, you are pulling air into your lungs, into these little sacs called alveoli. And those little sacs are covered in capillaries. That's what those little purple lines are. And in those capillaries, gas will run right through the single cell of the capillary barrier, and it will enter your bloodstream, and it will enter your red blood cells just by diffusion. So every time you breathe, you're pulling in air, and the oxygen is moving from high concentration in the air, high concentration in your lungs, into a lower concentration of oxygen in your blood. So it is passive, it doesn't take any energy, and it's totally dependent on the oxygen concentration of the air. So if I were to take this room and I were to decrease the oxygen content so that it was equal to my blood, I would no longer have oxygen entering my bloodstream when I breathed. So it's really just a concentration gradient that's allowing oxygen to get into us. The same is true about carbon dioxide. We produce carbon dioxide as we make energy in our cells. And so the cellular concentration of carbon dioxide is higher in me because I'm making it than it is in the air. And so that carbon dioxide is diffused into my bloodstream, and my blood will go into my lungs in these little capillaries. And from there, the carbon dioxide will exit, because of its concentration gradient, exit my blood into the air so that I exhale it when I breathe out. So high concentration of oxygen in the air, low concentration of carbon dioxide. In my blood, high carbon dioxide, low oxygen. And so we're net exchanging both of those passively. It doesn't require any energy just because of the concentration gradient that we're in. We're also going to use that concentration gradient to move the carbon dioxide and the oxygen throughout our body. So here's a nice cardiovascular system. We have your heart in the center. These diagrams like to show blood as red when it is full of oxygen and blue when it's short on oxygen. Your blood doesn't actually change color. It might go from like bright apple red to kind of duller burgundy, but it never turns blue. So that's just the way that we visualize it. Blue blood is not a thing for humans. It's always red. So here we have your heart. Your heart's goal is to pump blood to your lungs and from your lungs to your whole body. So your heart will beat. The blood will be pushed through your lungs, through these capillaries. It will pick up oxygen. It'll go back to your heart, and then it will get pumped around your full body. That's what this is trying to show you. Is up here are all the blood vessels that would go to your head and your torso and your lower half of your body. After it goes to your body, you're going to have exchange in the tissues. So in the tissues, carbon dioxide will enter or leave the bloodstream. Oxygen will enter or leave the bloodstream. And that will continue as your blood moves along its circuit. And then by the time it gets back to the heart, the concentration of oxygen has been used. It's been pushing those oxygens passively into the cells that need it. And carbon dioxide has been sucked into the blood from the cells that have been making it. And it goes back to your lungs, where carbon dioxide will follow its concentration gradient into the air in those alveoli air sacs, and then exhaled by you, where oxygen does the reverse. It is sucked into the blood from those air sacs and then is used in your cells. So in each of these, what should we be looking for? We're looking for the different concentrations of carbon dioxide and oxygen. That's what the P is, different concentrations. And you will gain or lose oxygen or carbon dioxide based on where there's more need. So here, your oxygen is going to be less than here. So oxygen is going to enter 40 and 100. And your carbon dioxide is going to be a little bit higher here. So it's going to exit. This is your air sac, your alveoli. Alveolus is singular of alveoli. Then going down to your body is going to get used in your leg muscles and your leg skin, in your knee joints, whatever you're doing. Here you can see that the carbon dioxide level is lower. And here it's higher. That's because these cells, your muscle cells, your skin cells, this is an adipose cell, that one's a neuron. These cells are producing carbon dioxide. So they're making more carbon dioxide than is in your blood. So it's going to exit those cells, get into your bloodstream. You have more oxygen in your blood than you have in these cells because these cells use the oxygen to make energy. And so the oxygen is going to go from high concentration to low concentration. It will exit your bloodstream. And what you're left with is less oxygen, more carbon dioxide, and you take that to your lungs, and then you have diffusion occur again. So it's a constant loop of diffusion with oxygen and carbon dioxide moving from high concentration to low concentration. So that's our respiratory and cardiovascular system. But diffusion is also really important for our digestive system. This is our digestive system. We have your liver, your stomach, your large intestine, and your small intestine. Back here, you have your other accessory organs, like your gallbladder and your pancreas. And the main goal of digestion is to break apart big molecules and use the nutrients and the energy in them to power our cells. So think about whatever you had for breakfast. Say it was a piece of toast. Those toast pieces are made of starches. Starches are glucose molecules that are stuck together. So I'm going to break the starch into individual glucose molecules. And now a single glucose molecule is small enough that I can absorb it into my cells. The cool thing about glucose uptake is it's controlled by hormones, which are produced by our cells. And the hormone that we use to control glucose uptake is called insulin. So insulin is made in our pancreas. The anatomy textbooks like to describe the pancreas as fish-shaped. So maybe you see a fish there, but here is the fish-shaped pancreas. It's right back here. It sits under your stomach, so it's right in the center there. And in your pancreas, you have several different types of cells. One of them is called the islet of Langerhans, which is just a great name. And inside there, you have beta cells. You also have alpha cells, but they do something different. Beta cells produce insulin. Now, we talked last week about how we start with DNA, go to a ribosome, use the Golgi apparatus, and then exocytose a protein like insulin. So we're going to do the same thing in this beta cell. The information to make the protein hormone insulin is stored in the DNA, but DNA cannot leave the nucleus. So you copy the information in mRNA. mRNA will go to a ribosome on the rough endoplasmic reticulum. It does it on the rough endoplasmic reticulum because we know we want it outside the cell. If we were going to use that protein inside the cell, we would just use a free ribosome floating in the cytoplasm. But we know we need to eject it. So we want to use a vesicle to eject it later. Thus, we do it on the rough endoplasmic reticulum because the ribosomes on the rough endoplasmic reticulum will put the protoprotein right into a vesicle so it can get transported. Once the protoprotein is created, it goes to the Golgi apparatus where it is modified and packaged and turned ready to work. And then it's put into another vesicle, and that vesicle fuses with the cell membrane and gets ejected. As you're ejecting it, you get a whole bunch of insulin that's just getting pumped into the bloodstream because all of these awesome beta cells all throughout this pancreas are pumping out insulin. Once the insulin is in the bloodstream, it will go to any cell that has a receptor for it. So it floods your whole body because your blood goes everywhere. And if your cell has a receptor for insulin, it will tell that cell, like this muscle cell here, hey, there's sugar, you should absorb it. And these cells will put up a receptor and they'll start to absorb the glucose. So the insulin tells cell to take up glucose. It's really awesome. If your cell has an insulin receptor right here, we often refer to these as ligand receptors, and the molecule itself we call a ligand. So if you see the word ligand in your textbook, that means that it's the messenger. And if it's a ligand receptor, that means it receives the message. So here's insulin that I just produced. It's a protein, but it's also a hormone. It's going to be received by any cell that has an insulin receptor. Once it's received by the cell sees that it got the signal that there's glucose in the bloodstream. And so it sends that signal to these vesicles that are waiting. These vesicles contain the glucose transporters, which we call GLUT4. That vesicle will fuse with the plasma membrane. And now I have the transporter for glucose in my plasma membrane, so glucose can enter the cell. When there is no longer a signal that insulin is there, these glucose GLUT4 transporters will be pulled back in. So they're only used when glucose is present. Here's a beautiful process of it. I want you to look at the steps and see if you can identify what's happening at each step. So the ligand, that is the message. Cell surface receptor, that receives the message. A secondary messenger is what is the response to a message inside the cell. Here you have a GLUT4 containing vesicle. It's going to fuse with the plasma membrane. That allows the GLUT4 transporter to be active. It will allow glucose to come into the cell. When glucose is gone, those vesicles will pull those transporters back out and they'll return to their waiting position until your glucose levels increase again. So I want you to think about diabetes now. What is diabetes? There are two types. There's type 1 and type 2. Type 1 is when your body does not make enough insulin. This can develop at any age, but it used to be called child onset diabetes because we were diagnosing it in children most. It can never go away. It's just a functional thing. About 5% of all diabetes cases are type 1. That means that 95% of all diabetes cases are type 2. Type 2 used to be called adult onset because it was diagnosed later in life. But now we know to call it type 2 because it can occur in children. That's when your body doesn't use insulin properly. It can develop at any age. And currently, the statistics are 1 in 3 people will develop type 2 diabetes in their lifetime, which is really high. So it's important to think about what happens when insulin doesn't work, especially if 33% of the population are going to experience this. I want you to imagine that you are a person with normal blood sugar metabolism, which means you do not have diabetes, and you just ate something that was super high in sugar, perhaps a donut and some cola. So I want you to think, what are your blood sugar levels going to be like right after you ate the donut and cola? And then what are your blood sugars going to be like several hours later? Think also, what is the insulin level going to be like right after the meal and several hours later? So here we have 3 people that had eaten a donut and a soda. We have patient A, patient B, and patient C. And we have 2 graphs for each of them. The first graph is blood glucose level from 0 to 300. And then we have time after they drank their cola and ate their donut in minutes, so 0 to 2 hours. The second graph on the bottom is blood insulin level from 0 to 500 and the same time range. So one of these patients has normal blood sugar metabolism. They do not have diabetes. They are healthy. One of these patients has type 1 diabetes, and one of these patients has type 2 diabetes. Who do you think is who? All right, the person in the center, patient B, they're the one without diabetes. They have a spike right after they had their sugary meal. And coinciding with that spike, there's an increase in insulin levels. Once the insulin is in the bloodstream, that allows those GLUT4 receptors to activate, and they will start to pull the sugar out of the bloodstream into the cells. As there is less sugar in the blood, the level of insulin decreases. So that is how it is supposed to work. Increase sugar, increase insulin. Increase insulin, decrease sugar. Decrease in sugar, decrease in insulin. So if you were to graph this every time you ate a sugary thing, it would go spike, drop, spike, drop, spike, drop. A person with type 1 diabetes, their sugar level elevates just like everyone else, but then it does not decrease. The reason it does not decrease is because type 1 diabetes are unable to produce insulin at healthy levels. And so their insulin level might spike a bit, but it does not have that giant jump like someone with a functional beta cells producing high insulin levels. Because the insulin does not increase drastically, the glucose is not taken out of blood. So for type 1 diabetic, you have low insulin levels and high blood sugar levels. For type 2 diabetic, the insulin is there. It has spiked, but the cell takes longer to respond or does not respond appropriately. So instead of this spike in insulin causing a crash in glucose, the glucose levels stay high. It's as if the insulin is being ignored. So a high level is produced, unlike type 1 diabetics, but it isn't having the same effect. And so you're up with high insulin and high glucose levels in the bloodstream. All right, so that was diffusion, osmosis, and glucose uptake. Email me anytime with questions, and I hope you have a great day.